Hello everyone, and welcome in this session on basic of bioinformatics operating using Python. Good morning, all of you. This session is going to start very shortly. Bioinformatics is the use of a computational approach to analyze, manage, and store biological data. The research in hello everyone, and welcome to this session on basic of bioinformatics operating. This session is good morning, all of you. Bioinformatics is the use of a computational approach to bioinformatics is the use of a computational approach. Hello everyone, and welcome to this session on basic of bioinformatics operating. This session is good morning, all of you. Just 1 minute. Okay. Yeah. Good morning. We are going to start the session. Bioinformatics is the use of computational approach to analyze, manage, and store biological data. The research in biotechnology, especially that involving sequence data management and drug design, occurred at a speedy rate due to the development of bioinformatics. A number of tools and softwares are developed for analyze and interpretation of biological complexity. There are a number of applications of bioinformatics with sequence analysis and alignment, molecular modeling, docking, annotation, and dynamic simulations to accelerate the biological research. It is expected that many features of bioinformatics innovation are likely to be stimulate analysis of a vast biological data. In this session, Dr. Praveen Kumar Ghotola will be the speaker. He has completed his PhD from NIT Rourkela, Odisha. Presently, he is working as a bioinformatics scientist in a Tata Medical Center, Kolkata. He has done a bachelor degree in biological and master degree bioinformatics and have good skill in programming languages such as C, C++, Perl, Python, R, etc. He has published a total number of 16 research papers in a reputed journal. He has also developed and got a copyright of 2 softwares, a graphical user interface for bioinformatics workstation, and second one is a software for tracing stem cell niche and lineage. His research area are cancer biology and stem cell biology. He has delivered numerous talks on national and international workshop training programs and conferences. I hope his expertise will be very informative to all the participants in this session. Welcome, dear professor, and now you can start your talk. I would like to first make host in this session.
Thank you, Dr. Meher, for the nice introduction. So if you make me the host, then I will start sharing the presentation. I'm audible, first of all. Is it clear? Voice is clear?
Yes, sir. You are audible and voice is very clear.
Okay, okay. So I hope all the participants are aware of all participants are some biotechnology or some other fellows are there from other background. Anyone?
Sit up. Sit up.
What did they say?
Sit up.
Sir, please check your. I have assigned you co-host, and I hope you can share your desktop. Please check it.
Hope candidates have background in biotechnology and bioinformatics. Stand up and listening.
Okay. Yes.
I give you below.
So I request to all the participants, please keep your mic muted. You can
okay,
if you have question. Please.
I started Okay, I started sharing my screen. Is it visible?
It is in the sharing, sir, but not visible yet now.
Okay.
Why is this taking time?
Yes, sir, it is fine now.
Is it
Yes, sir, visible.
now? Okay. Good morning. Okay, thank you. I'll start my presentation now. So, okay, good morning, everyone. Okay, I hope everyone is fine. Everything is good from your side. So today I'm going to give a small introduction on bioinformatics and how it is useful. Nowadays, if you take bioinformatics, this is an emerging field because there is a lot of genomic data, proteomic data, which is coming from different laboratories. Okay. So there is a need of bioinformatics to study the function of genes or whatever. So suppose, so without bioinformatics, as I'm a bioinformatician studying computational biology, so I'm more fascinated about bioinformatics because what I can say is without bioinformatics nowadays, biotechnology is okay, but biotechnology gives more information for that, so that it will make easy for experimental biologists who are practicing some for in future. So this bioinformatics is mainly nowadays an emerging field. So I think like people have started from different fields like mathematics. Mathematicians can also study this bioinformatics as computer science department people who are good.
Well, that last week Saturday, break now.
Sir, you are not audible. Please check your voice.
Yes.
Yeah.
Is it visible now?
Yes, sir, visible.
Okay. So I'm going to start my presentation. In between, if you have any doubts, you can interrupt me and you can ask. No problem. So coming to the definition of bioinformatics, this is an emerging interdisciplinary research area. It deals with mainly the computational management and analysis of biological information. So simply speaking, it is analysis of biological information using computational techniques. Like there are so many different programming languages which we can use and study and analyze this information. So I will not take much time as I have to give some demonstration on Python. So I will complete it quickly. So in between, if you have any doubts, you can interrupt me. So these are the different programming languages which we can use to analyze the biological data. So as everybody knows, R, Python, Perl, MATLAB, bioconductor packages, Galaxy, these are the things. So there are the basic programming languages which starts from C. So this R, Python, Perl, R are like the syntax which we write, programs which we write are similar to that. But R and Perl are widely used in bioinformatics because in R there are so many bioconductor packages which we can use. So for every analysis, there is different bioinformatics package. See, suppose if you want to analyze genomic data, there is different package. Suppose if you want to analyze microarray data, there are different packages in bioconductor. So based on what we want to study, based on that, we have to select our programming language because all things can't be done in R, all things we can't do in Perl, all things we can't do in Python. So based on our application, what we want to study, we have to select the appropriate programming language and we have to study accordingly. And if you are good in the basics, then it's not difficult part to write scripts or programs. So it is easy, but starting is a little bit, suppose if you are from a biotechnology background, as I'm from a biotechnology background, initially I also faced so many problems and writing scripts and using those interfaces. After, if you are habituated to that, then I think you will enjoy the programming. So you will feel very easy to use that, and you will become master in that, how to analyze. So I suggest, like before starting, you at least you should have some basics about programming on C, then it is enough, because all syntax are derived from there. So small changes will be there. Suppose if you see in R, if you want to write one program using loops, so we have a different syntax for that. Suppose if you want to write a program in Python, there is a different syntax for that. So it is not completely different actually. Small changes if you make, then you can understand and you will know how to write the program and you will know how to analyze the data. So this is the overview, like what are the different programming languages are there and how it can be used. And nowadays mostly people are focusing on Python because it is having so many applications and it is emerging, more emerging now when you compare with R, Perl, MATLAB and all. So Python is having so many applications. So suppose if you start digging deep into that, then you will understand exactly what it is and how can you use it for bioinformatics. So coming to the core bioinformatics, what it exactly is, it will start from the sequence, whether it can be a protein sequence or whether it can be a gene sequence. So the major starting part is the sequence. So once we get the sequence, what we can study, suppose if you get a sequence from there are a lot of things which can be done in that. Suppose if you gave you a protein sequence. So what exactly we can do as a bioinformatician, suppose we will check whether there is a 3D structure for that or not. So as everybody knows that 3D
Hello.
Yeah, is it visible, sir?
Sir, it is visible, but sometimes your voice is breaking. Please fix the problem if it is possible for you.
Okay, yeah.
Yeah, please fix the problem. I think
Actually, the internet
change
is
the
a
place
little bit
for network.
unstable.
Yeah.
Yeah.
Maybe it's network.
Yeah. So is it visible now? Is it audible?
Yes, sir,
I'm
visible.
audible now? Yeah. So hopefully, I think this is the unstable connection. So because of that, actually, now
Yeah.
I think it will be not a problem. So, so where yeah. So every if you want to analyze, everything starts from the sequence. So the sequence in the sense like whether it can be a DNA sequence or a protein sequence, so 3D structure. So using some bioinformatics techniques, we can predict the 3D structure of proteins. So there are different soft find out domains in that, like, and you can find out the promoter sequences. There are different. So what, like, I just want to say how it is useful. So every bioinformatics starts what you show or what you give them is analysis part. So we can also study different types of pathways also using bioinformatics. Suppose if you are given some list of the genes, so you can find out different pathways and how they are participating in the disease mechanism. All these things we can know. So suppose if you understand the pathways, then we know which gene we have to target and which gene is more participating in the disease. All these things we can definitely understand by analyzing these pathways, these things and all. So bioinformatics can be looks easy actually, but when you go depth, so it is little bit, it will create an enthusiasm that you have to do more. So this is the basic, like, what is available. So suppose, as I told, first we'll get a sequence, then from the sequence we have to analyze the data and we have to generate different types of models. So suppose if you are working protein sequence, if you are working on a protein sequence, then we have to model the structure, like 3D structure we have to design. And suppose when we are doing a wet lab, suppose if we want to determine a 3D structure, we will use some different techniques like X-ray crystallography and NMR. So these things, like, it is a little bit time taking, and sometimes the success of experimentation is we don't know whether we will get success or not. But using bioinformatics, we can predict the structure. So that is a prediction. So that prediction can be useful for wet lab scientists. So they will get an idea, like what kind of structures or secondary structures are there, like whether it is a alpha helix or beta strands, all these things. So coming to the basic concepts, using bioinformatics, we can study the evolution, protein folding, and protein function. Evolution in the sense, suppose if you have 2 to 3 sequences, suppose if you want to know how close they are, then we will go for phylogeny study. So that is called molecular phylogeny, and there are some evaluations. So we will come to know what of the like how close. Suppose if you are given gene sequences, suppose if you have 10 gene sequences, we don't know how much similar they are. Suppose using this molecular phylogeny or evolution, we can find out how close they are, which sequences are getting clustered, and what is their evolution history. So coming to protein folding, as I told, suppose if you have a protein sequence, we can model and we can predict the protein fold of that, and we can also predict the function of that. Using bioinformatics, we can also build some mathematical models, like mathematical models in the sense, this is some other field. So mathematical modeling of biological systems. So this is one type of interdisciplinary area. As I told, mathematical modeling means we can integrate some mathematical equations also in that, and we can model the biological mechanism, anything. Like suppose if you want to model cancer cell proliferation, using mathematical models, we can just model and we can predict how it is behaving. And we can identify different biological complex mechanisms in that disease or whatever. So this is how information processing in cells happens. So suppose if you take a nucleic acid, so there will be coding regions, regulatory sites in that. From coding regions, we will get transcripts. So the coding regions in this, those are proteins like which is mRNA and mRNA to protein. So this is the basic machinery, how it happens in the cells. So each thing, see, suppose if you see the regulatory sites, we can find out some of the domains in the sequences. Suppose if you see the coding regions, so we can identify what are the transcripts and which transcript can be coded into proteins. So again, different types of data we can get from a cell. So like suppose if you want to deal with genome data, we can get genetic data. Suppose if you want to deal with transcriptomics, then we can deal with mRNA data. And if you want to deal with proteomics, then it is a proteomics. So this is the thing, like how cell will generate, select and which data from be important. So based on your data, you have to select your analysis points. And as I discussed with you, like there is another interdisciplinary branch called systems biology. So in this systems biology, we can model whole system actually. So suppose if you take a living cell, suppose if there is any change from outside environment, how it responds and what response we'll get. So based on this, like we can completely model that mechanism, and we can understand exactly what is happening, how cell is responding to that particular perturbation or mutation. So these are the things which we can get. But the basics for all these things are bioinformatics. So everything starts with bioinformatics. From there are different types of branches which we have to study based on our analysis. So again, this is a small example which I am showing. How cell behaves with the help of external environment change. In internal environment, how changes will come and what are the different types of proteins it will get expressed, and how some pathways will change and how it responds. So this is an overall overview how one bioinformatics, like how bioinformatics looks. So it is like it is a hub of different sciences. Like suppose if you see genomics, as I discussed, mathematics, computer science, biophysics deals with modeling. Molecular evolution deals with phylogeny. Molecular biology, suppose see in molecular biology, if you want to, suppose if you want to if you want to do run a PC,
Sir, your voice is again breaking. Dr. Praveen, your voice is breaking. Please check it.
If you want to get gene sequence, based on that, we will design using bioinformatics. There are some, we can do some analysis on that, and we can Hello. Hello.
Sir, you can share your PPT with other one, so they can show on the screen, and you can only speak. So this solve your internet issue. Like if your internet is of a small power. Thank you.
Is it visible now?
Not yet. Yeah, fine, sir.
Yes.
Yes,
Okay.
fine.
So, so this is a overview of the bioinformatics. So what are the different branches are involved in this? As I discussed with you, like genomics deals with genome data, and molecular biology. Suppose if you want to design any primer sequence, you can design primer sequence using bioinformatics. Suppose if you want to model the protein structures and all, you can study biophysics. So suppose if you want to study molecular phylogeny, then you can deal with molecular evolution. So these are the things which you can study using bioinformatics. So it is a merging of different interdisciplinary areas. If you have any doubts, you can ask me. Hello.
Yes, sir, you are audible. Please carry on.
Yeah, okay. If you have any doubts, you can ask me, no problem.
Hello?
Yeah, tell me.
Yeah, the biophysics you were talking about, I don't really understand.
Which one? Sorry?
Biophysics.
Okay, yeah, thank you. So, like biophysics in the sense, suppose if you have any protein sequence. So basically what we will do is, suppose if you have a protein sequence, we can study the protein domains and we can study protein function from that. So biophysics deals with mainly, suppose if you are a wet lab scientist, suppose if you are working on a protein sequence, suppose if you are working on a proteomics. So you want to determine the 3D structure of that protein. So 3D structure in the sense, like we will use X-ray crystallography or NMR for determining the 3D structure. How bioinformatics can be useful is like you can simply get this protein sequence and using some softwares. Like there are some softwares like Modeler is there, Swiss Modeler is there. So these are the softwares which we can use and we can predict the 3D structure of proteins. So biophysics in the sense, so suppose if you take a 3D structure, in 3D space, it will create different, suppose it will create some conformations. So we can study that conformations using molecular dynamic simulation. So that is one area which you can understand. Suppose if you take a protein and put in some solvent, obviously there will be some changes in conformation. So using that, by using bioinformatics, you can study those conformations. Those conformations are called as molecular dynamic simulations. So this is called as biophysics in the sense, like how it can be useful.
Okay,
hello?
thank you.
Okay.
Thank you.
Hello, good morning.
Yeah,
Good
yeah,
morning.
yeah.
Good morning.
Yes, good morning.
Good morning. Now, how does bioinformatics relate with legal? Because I see ethical, legal, and social implication, but I'm particularly about the legal aspect of it. How do we use bioinformatics in legal implication?
Sorry, actually, it is not clear. Can you comment? Can you speak it again?
I said I saw the bioinformatics in context, and I saw ethical, legal, and social implications.
Okay,
and I want to know how bioinformatics is related to legal. Are you not talking about legal issues? How is it related to legal bioinformatics? Thank you.
Yes, okay. So ethical issues in the sense is sometimes what will happen, You see, experimentations will generate a data. Okay, some people will make it public. Suppose if I want to work on some genomic data, suppose let's talk about cancer. Okay, so there it will come exactly what ethical and social implications will come. Suppose if you are working on a cancer biology, what exactly happens is you can't work on patient data without their consent. Obviously, you will take consent from them. Without consent, if you use their data, patient data, obviously, if I publish some paper, obviously ethical issues will come. Suppose if I am not taking any ethical permission from them, they have a right to charge me legally or whatever it is. So that is what ethical, legal, social implication comes. Like, suppose if you are working on a data, bioinformatics itself is working on a data. So sometimes what will happen, you will get publicly available data. That means they are making it public. So you have a right to work on that. But suppose if you take a cancer study, cancer is a cohort study actually. Suppose if you are working on such a big cohort without permission or without consent, you can't work. Suppose if you are working, they will obviously they will charge on you. So that is what exactly ethical, legal, and social implication means.
Okay, thank you very much.
Is it clear or?
Yes, it's clear. It's clear. It's clear.
Yeah, thank you. So let's.
Thank you.
So today, next I'm going to discuss about some biological databases. So database in the sense, how we will get the data. So what are the different types of resources are there which are available, and how we can get that and how we can use for our study. So there are different types of biological databases for that. Suppose if you see this picture, in 1995, the number of genes in the database are getting increased. Like, and at that time, like the number of genes in database are increasing so much, less than publications actually. So suppose if you want to get the data, there are different types of databases, biological databases, and they categorized into different ways. So primary data, secondary data, and tertiary data. So primary data deals with the sequence. So that sequence in the sense, whether it can be a gene sequence or protein sequence or whatever it is. Suppose if you see, so there are different types of databases which I will show. So in your free time, you can just browse there and you can see what information you will get. Secondary data in the sense, the information we'll get from primary data. Suppose if you take a protein sequence, suppose if you take any gene data, you will get a motifs, regular expressions from that. So these things can be stored in secondary database. So those are called secondary databases actually. Next one is tertiary data. So tertiary data in the sense, suppose if you take a protein data, 3D structures, suppose if you are dealing with some 3D structures of a protein, in that actually what will happen, they will store the 3D structures of the protein sequence, like which there are already determined 3D structures using experiments. So that is called tertiary data. So tertiary prediction data in the sense, those are 3D structures. So suppose if we take an example, PDB database is there. So that is a tertiary protein structure database. So these are primary biological databases. Suppose if you are dealing with nucleic acids, then we will go with EMBL, GenBank, DDBJ. So these are totally deals with nucleic acids. So suppose if you are working on any DNA sequence or RNA sequences, then you can use this. So EMBL means European Molecular Biology Laboratory, GenBank, DDBJ is. Suppose if you are working on protein sequences, so there are different types of databases like PIR, Protein Information Resource, FISPROT, Tremble, NNR3D, all these things. So basically, primary biological databases deals with sequences. So these sequences can be categorized into different ways like nucleic acid, proteins, all these things. So this is how the information flows. EMBL, as I discussed earlier, like it is Europe. So GenBank, it is maintained by NCBI, USA. DDBJ, so that is DNA Data Bank of Japan. So it is headquarters is in Japan. So this is how they are maintained. So different databases from where they are and how they are maintained. As I discussed, this is a GenBank. suppose if you go and type GenBank in your web browser, you can search any gene there. So there are different types of genes. Suppose if you type the gene name, so this is what you get from there. So you will get all the information about what sequence it is, and what kind of sequence it is, and what exactly it is, how, whether it is a coding sequence, whether it is annotated or not. If it is not annotated, then using some bioinformatics techniques, you can annotate that. So you can simply type GenBank in your web browser and you can see exactly what you will get. You can take any gene. There are so many genes. So suppose if you type that gene name, this is what the information you will get exactly. So another one, SysProt. So this is protein sequence database. Suppose if you want to work on proteins, so SysProt gives you complete information exactly what it is. Again, it is file format, how it looks, and what are the different types of things we'll get from there, all these things. So other primary protein databases are there, as I discussed. Tremble is one, that is similar to SysProt. And SP Tremble is another one. Suppose if you want to study on immunoglobulins or whatever it is, so you can use REM Tremble. So these are the different types of protein databases which are there actually. So if you want, I can share the slides. Then you can understand exactly, and you can browse it yourself, and you can understand what exactly it is. So quickly, then I have to show some other research, how it is, like how I used in my research, so that everyone will get an idea. So I will share another screen. So this is my research area actually. So what I studied is detailed study on molecular mechanism of pluripotency and reprogramming in testicular stem cells. So this is my research actually, what I have done exactly. See how you can use it, like what type of data and what type of methodologies are used. So I will just give one small overview so that it may be useful for your research if you are designing some research projects. So first thing is like, what are testicular stem cells? So testicular stem cells are self-renewing population. These are present in testis actually. So main advantage to study is this can be used in the treatment of male infertility. So nowadays male infertility is a major problem which we have to address that. And spermatogoneous stem cells which are present in our testis have the ability to differentiate into sperm. Suppose if we isolate these stem cells and put it in some graft, then under some culture conditions, so it will generate as, it will differentiate into sperm. And this can be used as a treatment in male infertility. So this is what actually, so when we are isolating the cells, so what are the different types of paths we will take? Like suppose if we take, when we are isolating the germinal stem cells, so it will take two paths actually. So it will give rise to sperm, it will differentiate into sperm, and it will give rise to germinal pluripotent stem cells. So these germinal pluripotent stem cells are pluripotent in nature, and it will give rise to the cells which arise from three germinal layers like ectoderm, endoderm, and mesoderm. So this is what, see, when we are isolating the cells, so it will take two paths. One is germinal stem cells, another one is germinal pluripotent stem cells. So these germinal stem cells, when we are culturing it, it will get
hello, sir.
Hello, yeah, tell me. Yeah.
I can't see anything. I can't see what you're displaying. I can't see.
Is it visible? No.
No, it's not visible. Okay, now it's visible now.
Yes, it's visible now.
Yeah.
It's visible now. Thank you.
Okay.
Maybe you start from the beginning so that we can get it.
Yeah. Okay.
Thank you.
Okay. So, so this is my research area which I worked on, like detailed study on molecular mechanism of pluripotency and reprogramming testicular stem cells. So this is just an overview. Suppose if you have a data, suppose if you are working on any cancer, suppose if you are working on any data, how to design your experimentations in bioinformatics exactly. So what kind of data we will get and how to design methodology to study or analyze. So these spermatogonia stem cells are present in the testis. So these can be used in the treatment of male infertility as they will differentiate into sperm. So this can be used in the treatment of male infertility. So suppose when we culture these germinal stem cells, so they will take two paths. One is they will differentiate into sperm. Another one is they will give some different types of cells called germinal pluripotent stem cells. So these germinal pluripotent stem cells will undergo some reprogramming and they will generate the cells from three germ layers like ectoderm, endoderm, and mesoderm. So my study is like how this reprogramming is happening. So how these GPS cells are coming from GS cells. Bioinformatics, using bioinformatics, how I have to answer this. So using high throughput sequencing data analysis only, we can computational techniques, we can study this and we can study the molecular mechanism, how it is happening. So these are the different types of objectives I framed and how I have done, like using machine learning, using some R programming, how I designed. So the different objectives are there, different methodologies I designed, and I completely used publicly available data, which is available in databases. So in my study, I used RNA-seq data, ChIP-seq data, microRNA data. Like I studied every sequencing data which is available on this both germinal stem cells and germinal pluripotent stem cells, and I designed some methodologies. Some are novel, some are existing ones, and I used some things and I generated a regulatory graph from all these things. This is a regulatory graph which I am showing is like how it programming is happening and what are the different programming, what are the signaling pathways getting activated, how. So this is about my research, like how you will get the sequence, like after getting the sequence, how you will, which programming languages you can use, and accordingly you can design your methodologies and you can study. So quickly, next I will just go to Python programming and I will show some things which you can do by it yourself. Okay, is it visible now? Hello?
Hello? Yes, it's visible.
Okay.
It's visible now.
Okay. So now I will show like how to use. Yeah. Hello.
I can see now.
Is it visible?
Yeah.
Yes.
Okay.
Yes.
Yes.
So this is now I am going to give small introduction about Python programming and how you can use this in bioinformatics. Like small things I will discuss. Suppose if you have a DNA sequence, how will you calculate the GC content from that? Some basics I will just show how to do. Okay, so as I discussed, this Python programming is high-level programming. So syntax allows programmers to express concepts in fewer lines. So when you talk about programming languages, suppose if you take C, C++, if you want to write a programming, suppose if you want to write a program for sorting two numbers, you have to write a big program for that. But when you take up scripting languages like Python, R, or Perl,
Signal is
within
poor.
less number of lines, we can write our code. Suppose if you want to sort a list of numbers using sort function, you can sort that. So that is the advantage when you are using these programming languages. But when you use Hello.
Hello.
Hi. We are not hearing you, sir.
Is it visible?
Visible. Volume is bad. Voice.
Signal strength.
Is it visible now?
Very visible.
For the audio. Hello.
Audio is bad.
Hello.
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 Yes, sir, it is visible, but sometimes your voice is breaking.
Oi. Rancoso. Isso foi comigo. Hello.
Dear participant, just please wait. He will join again. Yeah, he is changing the system.
Okay.
Yeah.
Is it visible now?
Yeah, it's visible.
Hello.
Yes, sir. Desktop is visible, but your voice is breaking.
Hello.
It's visible now.
Sorry for the interruption. Actually, internet connection.
Maybe network.
Yeah, actually, internet is not stable. Actually.
Okay.
Is it visible now? I'm audible.
Yes, sir. Please. Yes, visible. It is visible. Fine.
It's
visible is it audible now? Is it audible? Am I audible?
Yes, sir. Audible. Please carry on.
Yes, audible.
Am I audible or not? Like, yeah. Okay. Yeah. So I will just show some of application in Python programming, how it can be used. So this Python programming is high-level programming. So its syntax allows programmers to express concepts in fewer lines. So suppose if you are using any C++ programming, suppose if you want to sort two numbers, you have to write a big code for that. But using scripting languages like Python, Perl, R, within single line, you can sort the list of numbers or whatever it is. So that's why it will decrease your line of codes, and it is having so many applications. So how to install this? Like, what I am using. Yeah, so suppose this is how the Python shell looks actually. Is it visible?
Yeah, it's visible. It's visible.
Yeah. So this is what actually after installing Python. So there is a shell called IDLE shell. Suppose if you type IDLE shell in your computer, then you click that one, then you will get this. So in this actually you can run your programs, whatever you want to do. See, suppose this Python, it can be used as a calculator. See, suppose if you want to add two numbers. So it can be used as a calculator. See, some basic operations if you want to do, you can do using this Python actually. So there are different types of applications are there. I will just discuss small things, because if you want to know in depth actually, there should be some separate workshop for this Python programming completely, so that I can be more specific what I have to teach. So every programming languages deals with variables. So suppose if you want to store any number or anything, that is called a variable actually. Suppose now I see I will store some variable here. Suppose if I want to store the value of 18 in a variable, then I will store like this. Suppose if I type X, then that variable will come like this. So this, suppose if you want to work on that particular sequence or whatever it is, you have to store that sequence or whatever in the variable. Then you have to do the operations on that. So I will just show some things like how to take a DNA sequence, how to analyze that sequence. So basically DNA sequences again deals with strings. So suppose if you take a DNA sequence, so ATGC, that is a string. So you have to know what are the string operations are to be done in Python programming and how you have to analyze. Now I will just show you how to store this string in in a variable. Suppose if I am taking a variable A here, so I am storing that value of string in that. Suppose if I type A, then that string which I stored can be, I can retrieve that by simply typing A. I will get that particular. Okay, in strings also you can simply manipulate these things. Like suppose if you want to change this into capital letters, you can do. Now I will just show some small string manipulations how you can use. Okay, so first what I'm doing is I'm taking one variable, so that is called signal peptide actually. So I am storing that. Okay, that variable. Suppose if I want to see what variable I have stored. So this is a variable which I stored. So signal peptide is my variable, and I stored some sequence in that. Okay, so if you see here, this sequence which I have stored is in upper letters actually. So suppose if I want to convert those into lower, simply what I will do is, there is a function for this. Okay, signal peptide is a variable dot lower. So this is a manipulation. Then what is happening is everything which is in capital is converting into small words. Okay, so there are different types of operations are there in strings also. So these are the operations which you have to use, and based on that you have to write your scripts. Okay, so one more operation is like replace. So in this replace, what will happen? Suppose if you want to replace any string in that, using that method you can do that. So I will show one more example. So I am taking one more variable that is called DNSSEC. Okay, so there I am storing I am taking a string, and there are 7 letters in that. Okay, so this is the string which I stored, like TTGCTAG. Okay, now suppose if you take, this is a DNS sequence actually. Suppose if you want to convert that into RNA sequence, what you have to do? Anybody? Am I audible?
Yes.
Yeah, can you please tell me, somebody can tell me how can I convert this DNS sequence into RNA sequence? So I have taken one variable here, that is DNSSEC. So I stored a DNS sequence in this, like TTGCTAG. Now I want to convert that into RNA-seq sequence. So what can be done? Anybody can say, like not in a coding view. Yeah, tell me.
Hello, can I say?
Yes, yeah.
You type RNA sequence.
Yes.
Am I correct?
No, see, DNS, I have taken the DNS sequence here. Like TTGCTAG is there, no? This is a DNS sequence.
Hello,
Yeah.
Somebody on the chat
Yes,
said. Somebody on the chat said you can change T to U.
Yes, yeah. Exactly. So suppose if you have the DNS sequence, okay, so that is a string. So DNS sequence, in the sense, in the place of thymine you need uracil. So in the place of T, U should come. Okay, how we will do this? Using Python. Okay, so now I am taking another variable. Okay, so mRNA-seq is another variable I am taking. DNS-seq is a previous variable. So in that we have DNS sequence actually. Here what I am doing is I am replacing P with U. Okay, dot replace. This is one operation. This is one what I can say, operation or string manipulation which you can use. Okay, now simply you can type this mRNA-seq. So if you see here, exactly what happened, in the place of T, uracil has come. So simply this is a replace. So using replace function, what we have done, we simply replaced thymines which are present in the DNS sequence. We have a big sequence, like obviously DNS sequences will be very huge. So then what you have to do is that so DNS sequence which you have to save that in FASTA format actually. So that FASTA format sequence, you have to read it, then you have to use this dot replace function, and you can simply convert the DNS sequence into RNA sequence. Okay, so one method which you can manipulate string is replace. So you can replace your old base with new base. So old base here is thymine, and simply I replaced with uracil. Okay, so another one. Suppose you take any DNA sequence. Okay, what will happen? Initially, you will see Yeah. Yes.
Hello?
Yes, tell me.
It's not necessarily it should be you. It could be any other of the DNAs, right?
No.
It could be any of the DNAs. It's not necessarily it should be you.
Hello?
I said it's not necessarily it should be you.
No.
It could be
Here
any
actually,
other, right?
See. No. See, here I am. Yes, tell me.
It has to be you.
Other. See, it is not. Here actually we are here I am just asking how to convert the DNA sequence into mRNA sequence. So obviously you have to replace T with uracil, because in RNA sequence you will have uracil, not thymine. Thymine is present in only DNA sequence. So simply here I am replacing thymine with uracil. You can replace anything with anything. Like, see, suppose if you want to replace T with M, you can do it. Suppose if you want to replace G with T, you can do it. Anything.
Okay.
But here actually I am asking you to convert DNA-seq to mRNA-seq. So DNA sequencing, obviously in DNA sequence you will have thymine. So in the place of thymine, in RNA you will have uracil. So here you have to replace T with U. Am I clear? Hello?
Yes, yeah, I'm okay.
Hello?
I'm okay. It's clear. You can go on, sir.
Yes, sir. It is now clear.
Yeah, okay.
It's
Please
clear.
go ahead.
Okay. So next operation which I am going to discuss is count. So like replace, there is a
Hello? Audio, please.
It's like his network has gone off again. No screen, no audio.
Hello?
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 Hello? Hello? Hello?
Hello?
Hello?
Hello? We can hear you.
Hello, it seems like we lost network. Whether his network has gone
His network is so bad. His network is so bad.
Yeah, it's fluctuating, I think.
Hello, am I audible? Hello?
Yes, yeah.
Okay, you're audible now.
Yeah, I am not able to share the screen. I don't know exactly what happened.
Okay. Network problem.
Is host there? Like I'm not able to share the screen.
Okay, just 1 minute, sir. So now you can share the screen.
Yes, yes. I don't know. In between, I don't know. Suddenly, screen sharing is offing. I don't understand exactly.
Sir, it happened due to the low network.
Okay.
Yeah.
The screen is on now.
Yes, sir. Screen is visible.
Okay, okay. So next operation which I am going to show you is, suppose if you want to calculate the GC content from the DNS sequence, how you have to do. So there are some, there is one operation called count. So using that, you can count how many times G is present in your sequence and how many times G is present and how you can calculate the percentage. Okay. So simply now I'm taking another operation. So my main objective is to calculate the GC content from that sequence. So here I'm using dnssec.count. That means it will count how many times C is present in your sequence. Okay. See now. So what it says is only one C is present in my sequence. Now similarly, I will just count G. So what it says is 2 G's are present. Now I have to calculate the percentage of this G plus C content. How will I do?
Another operation.
Okay. So what exactly it is? So first what I have done, I have taken the DNS sequence, then I counted how many times this C is present in the sequence. So what it says, one C is present in my sequence. Another one, how many times G is present. So 2 times is there. So now what I am doing, C plus G means 1 plus 2. Okay. By length of DNS sequence means total length. So here total length is 7. Okay. 7 bases are there. TT, GC, TH into 100. So the GC percentage in this sequence is 42.85%. Like that, you can calculate your GC content for a large FASTA file also using these small operations. So simply what it does, dnssec.count. So .count, it means like count how many times C is present. It count how many times G is present. So these are the things, basic operations are how you will do using Python programming. Okay. Suppose if you want to find out the AUG, AUG is like codon, which your protein coding region starts from AUG. Suppose if you want to find your codon, you can use this string operation find. So using that find, you can I will simply show how to use that. Okay. Suppose if you see here, mRNA-seq is sequence which I converted DNA to RNA-seq actually. So now simply I am asking to find this particular sequence where it is present. Okay. So because in RNA sequence, the coding region starts from AUG, that codes for formal methionine actually. So suppose if I have given a RNA sequence, if I want to find that starting coding part, so using find, I can do that. Okay. So these are the basic operations which you can do using Python programming. Okay. So suppose if you get the sequence, initially you can convert that sequence into RNA sequence. You can convert that DNA sequence into some other sequences. And suppose if you want to know the GC content, you can calculate using this Python. So these are just, I gave you the overview, but there are many of operations in this Python actually. So what I suggest is just start using that. Suppose if you are okay with that, then suppose if you feel it is, I want to go with more depth, then you can study more about this Python programming. Suppose if there is some specific workshop or particularly bioinformatics analysis using Python, then I think definitely there are a lot many things which you have to learn, so that you can use for your research actually. So this is what I want to show. Suppose if you have any doubts, you can ask me whatever, like regarding any research thing or whatever. If anybody have any doubts, you can ask me, like regarding any in-depth bioinformatic thing. It's like anything you want to discuss about your research. Suppose how you want to use for, like if you have any questions, then I can answer those.
Yes, dear participant, if you have any question, please feel free to ask. You can unmute yourself, or either you can put your question in the chat box.
Okay, there is one question.
There is a question in the chat box.
Okay, yeah. Sir, I want to do MSc in bioinformatics. Okay. Lovepreet Singh. Okay. So, okay. Actually, I don't know what bachelor's, like your bachelor's is from. Like, can you unmute yourself?
Lovepreet, you can unmute. Please feel free to ask. Lovepreet Singh, you can unmute and then ask the question.
Hello.
Yes, yeah. Yeah, tell me. Hello.
Hello, sir. I am currently doing my BSc in biotechnology.
Okay.
And I want to do my MSc in bioinformatics. So what should I do? And can you please guide me?
Okay. So, okay, actually, if you are from a I am also from same background actually. My BTech is from biotechnology. Later, my master's is from bioinformatics. So you can, if you want to do bioinformatics, then yeah, I will definitely suggest you to do. But what is my suggestion is, suppose if you want to know, what kind of study you want to do. Like suppose if you are going to bioinformatics, obviously you should be good in some programming languages, like whether it can be a Python, R or Perl, anything. Suppose if you are clear with your basics in your bachelor's itself, then when you are taking this master's course, it will be easy for you actually.
Sir, I have done basics in HTML and Python.
Okay, then yeah, if you are good with your basics, then I think yeah, definitely it will be easy for you, because in say curriculum, bioinformatics curriculum will have so many things, like biological networks will come in that, mathematical modeling will come. Suppose if you are good in at least some bioinformatics presentation, at least some things, then it will be easy for you. Okay, but my suggestion is, increase your programming skills. Okay.
Okay, sir.
And at least you should know all basics. See, suppose if you can take any programming, suppose if you want to go for Python, you can go do in-depth Python programming. But see, using Python completely, you can't do complete bioinformatics. Okay.
Okay.
Why I'm telling, because everything for on one study you have to use some different programming. So you have to be good in basics, R also, Perl also. Based on your study, based on what you want to do at that particular point, you have to select your programming language and you have to do. Okay. So that is what. And there is one more question, like the last code in Python was not clear. please repeat dot find again. Okay. Okay, I will just repeat that again. Okay. So find. So here dot find means, so suppose if you are taking some group of characters. See, suppose if you take any regular expression in the DNA means some sequences will be similar, obviously. See, suppose I want to say GATC in that sequence. That sequence will be very huge. Okay. I don't know at where that GATC is present in that sequence, because by naked eye I can't check that. Okay. So what I have to do is, using some bioinformatics techniques, I can do that. So suppose, obviously DNA sequence, RNA sequence are strings. So you have to use some string operations. Here, dot find, using dot find, you can find out that particular group of characters position. Okay, at that what position it is present. See here, I will just show one more thing. See, suppose if you take that sequence, suppose if I want to find TTG, okay. UUG. Okay, see, suppose if you see this, okay, mRNA find mRNA-seq dot find UUG. I am asking where that UUG is present. So what it is showing, see, suppose if you take UUG, it is telling that it is present in 0. Okay, 0 means like every base address in the programming languages. Now, suppose if you take arrays or whatever, it starts from 0. So 0 will be your first position, 0, 1, 2. See, here I am asking where this UUG is present in my sequence. So mRNA sequence I am searching, and I am asking where this UUG is present. So what it says is it is present in 0th position. So suppose if you see that it is present in 0, so UUG. So that 3 group of characters is present in 0th position. Okay, like that. Suppose if you want to search for AUG in the sequence, so it will show you exactly at what point, at what position, at what base address that AUG is present.
Hello?
Yes.
Why is it 0? Why
Sorry?
is it 0? Why is it 0?
Sorry, I didn't get you.
I said, you said UUG.
Yeah.
And you put 0 here. Why?
See, UUG is like, I want to find the position of this 3 group of three sequences which are present in the sequence. See, suppose my in my mRNA sequence, what is the mRNA sequence? UUG, UUGC, AG. Okay. Now I want to see at what position this UUG is present in that mRNA sequence. So what I am doing, mRNA-seq dot find UUG. So what it's showing is 0 means it is present at 0th position. Okay. That is what I'm telling. In arrays or any string operations, in programming languages, your base address will start from 0, 1, 2, like that. So here UUG is present in 0th position. Suppose if you take GCU, it is present in see here. Did you find the difference between these two things? Okay, yeah, UUG is present in 0th position. So GCU. So this UUG, GCU are present in the my mRNA sequence. So I want to know at which base address it is present. Suppose if you take the sequence here, see here. Based on the address, see, suppose, as I told you, it will start from 0. 1, 2. So that's why 2 is 2, why it is showing 2 here. Exactly, this G is starting from 0th base address. You understood, no? Exactly what I'm going to convey you.
Okay.
Yeah. So this is what actually the base addresses in programming languages like arrays. If you take strings, if you take it will start from 0. So the first letter which you are seeing, it is starting position is 0 actually. The next one is 1, 2, 3. So it will not start from 1. So here I am just asking the position at which where it is GCU is starting. That means it is showing the sequence. See, if you, there is UUG, no. So first 0 is U, second 1 is U, 0 is G. So that's why it is showing you 2. That means this GCU is starting from second base address. Is that clear?
Okay.
Yeah.
Okay.
So if any doubts are there, you can ask me. If any questions.
Hello.
Yeah.
Good morning.
Good morning. Yes.
Yes, I want to ask. Suppose someone have background in biochemistry, then in mathematics, and also mathematics.
Yes.
Can one do a postdoc in bioinformatics, even with a little background of Python?
Yeah. See, I don't know. Like, yeah, have you done your PhD?
Yeah.
PhD in PhD in?
PhD in mathematics.
Okay, mathematics. Okay, yeah, good. Yeah, now you want to do your postdoc in bioinformatics. That is what your question, no? Hello?
PhD in Hello?
Okay, no, now like how you want to how you want to okay. See, actually, anyway, you have done your PhD in mathematics and you want to move to bioinformatics. Actually, as a mathematician, you have so many opportunities in bioinformatics actually. So that is what I'm telling. Suppose if you want to move to bioinformatics, there is one branch in that, mathematical modeling of biological systems. Okay. So yeah, this mathematical modeling of biological system means that you can model particular biological process using mathematical equations. Okay. See, suppose there are some differential equations. No, you can model using differential equations also. Calculus, integral calculus is there. So you can model your biological systems using mathematical equations. So mathematical modeling of biological systems is nowadays this is becoming very important field in bioinformatics. Suppose that is not a difficult actually. Suppose if you know how to how bioinformaticians and biologists will feel, you know, when we are moving to mathematical modeling. So we feel difficulty because we don't know that much mathematics. So suppose if you ask me to write one differential equation, it is difficult for me. But for mathematicians, it is very easy. But what thing you have to understand is you should understand biology actually. Then you can convey that in forms of mathematical equations. Okay. Suppose if you have a good mentor who is teaching you biology, he make you understand exactly what you have to convey using mathematical equations, then it will be very easy for you. And it is not a difficult part. But yeah, if you want to move, if you want to move,
I have a background in biochemistry, like protein sequences and all that. I have a PhD in biochemistry.
Yeah, then it is very easy. It is not difficult for you.
Okay.
So it is very easy for you. Don't think like it is you are moving completely in different area. But mathematical modeling of biological systems is suitable for you actually.
Okay.
So that is called systems biology. Even biochemistry and biochem also there in that. So it's not a difficult part.
Okay. Thank you.
Yeah, thank you.
there are many mathematicians who are working on biomathematics, biomathematical modeling, and also bioinformatics.
Yes, yes.
So it is very quite easy to move for bioinformatics, biomathematics, biostate for mathematicians.
Yeah, you can get my contact information from host actually. If you want to contact, if you have any doubts.
Yeah, Professor Patience Ivan is asking about contact number. Yeah, I will provide you. He is regularly attending our workshops and seminars. There are some participants who are attending this session through live streaming on YouTube, and there are some questions from the participant.
Yes.
We will discuss one by one. They put their question on the chat box. Just 1 minute, sir.
Actually, I'm not seeing anything on chat box.
These are the questions on the in the chat box on live streaming YouTube.
Okay.
Yeah. Just 1 minute, sir.
Yeah. Can you please share those questions, because I'm not seeing anything.
I would like to share with you. Yeah, just 1 minute, sir. Yeah, I think my desktop is visible to you.
Yeah, it is visible. Yes.
Yeah. So the question is the Kennedy Kudo is asking, is there any manual for Python programming?
Yes. Yeah, I have a manual with me with the Python programming, and some operations are there. Okay, I can share with the host if they want, and yeah. But what I'm expecting is like before sharing a manual or something, like what I feel is like you should be good in some basics of Python programming. At least you should know what you have to do, exactly what you have to analyze. Okay, and yeah, if there is some specific training program or workshop on particularly, see, like bioinformatics applications in Python programming, analysis of biological information using Python programming, where we can go in depth from the basics. So that will be like a nice platform to learn actually. So we can have more sessions on particularly only on coding, how to write a big codes and all, and we can give some live demonstrations. Okay, this is just an introduction, like how it can be used, like how we can use that Python programming. So definitely I'm looking forward if there are some workshops which are going to be conducted. Is there any particular programming program for this bioinformatics using Python programming? I'm always there to help you. So no problem. Yeah, if anybody needs guide Python programming for bioinformatics, I can provide. I can just send it to host. So he can share with you all people. No problem.
Yes, sir. We have already organized several workshops on R programming and Python. And in the coming days, also there will be several programs, 1 week programs. And also very soon we will organize 1 week or 2 week training workshop on bioinformatics from basic level to advanced.
Yes. Obviously, that will be very useful for
yes, because this is the emerging area and a lot of importance in the research. Sir, there is one. Yes, there is one more. Sir, there is one more question.
Yeah.
Precious Margini asking, he is microbiologist and wants to do master in bioinformatics. Is it possible for her?
Sir, yeah, like see, if you are studying any bachelor's degree, particularly on biotechnology or some life science related, if you want to learn, if you want to move your background to bioinformatics, yeah, definitely you can do. See, microbiology also have some role actually. Suppose if you want to microbiome data analysis is there. If you want to study particularly microbiome which is present in the gut, obviously bioinformatics will help you. Yeah, you can do it. It's not a, it is a, yeah, it is not a, it is a good option actually. I'm not telling you can't do, but you can do definitely. Because your masters will decide your PhD journey. So whatever you are studying in your masters, you have to take out towards your PhD. Suppose if you are interested to do PhD in bioinformatics, then if you are doing masters in bioinformatics, then it will be good. And the advantage of wet biology and wet experimentation techniques and this bioinformatics is people who know both wet lab and dry lab will be very good, and they have more opportunities than others. So yeah, you can do it. It's not a bad thing.
Hello?
Yes, sir.
Hello. Thank you very much for this presentation.
Thank you.
I personally enjoyed it, and it's very, very interesting.
Yes.
I just want to ask that, is there any software package for bioinformatics?
Yes.
Is there any software package?
Yeah, see, that is what I'm telling, no? Like, suppose if you take R programming, okay, bioconductor is one, okay? Bioconductor package is there. In that, there are different types of bioinformatics packages are there. Okay, R programming, bioconductor. Okay, so you can simply type, if you want to know more, you can simply type bioinformatics packages using this bioconductor in R. You will get so many packages, and there are very less number of packages in Python actually. Nowadays, actually, people are using Python in bioinformatics also, and they are developing some packages. But most packages, what you will find in internet are all are like this bioconductor packages which belongs to R programming. There are so many. So you have to see based on that. See, suppose if you want to analyze DNA-seq data, there is one different package for that. Suppose if you want to study RNA-seq data, there is different package for that. So based on your study, you have to select your package. And in R programming, you will have manuals actually. So suppose if you have basics, you can see that manual and you can do your programming. No problem, not a problem. There are so many packages.
Sir, also MATLAB provide bioinformatic tool, and I think this is very useful for all the.
Yes. Yeah.
Yes. Any more question, please, from participant side?
See, webinars are like, if you have any doubts, you can express. Like, suppose some people will be stuck in their research. Some people don't know how to use bioinformatics in their research. Suppose if you have any questions, it is a nice platform. I can give some suggestions, and yeah, definitely I can help you if you are stuck in somewhere.
So how do you get your contact? How do we link you up?
Sorry?
How do we contact you?
Yeah, I will provide my email ID.
We will, any participant, yes, sir. I would like to explain. Dear participant, if you have any question or any need in the future, you can contact us. I will provide you also the email address of the professor so that you can easily contact to him.
Can you post it? Can you send it to our chat page? Hello?
Yes, please.
I said, can you send to our chat page, the link, the contact?
Yes, I will send you. Yeah, I will send you.
Right. Okay, sir. Thank you very much, sir.
Thank you. And there is also one more question from Mathematic Arena. He is asking, can I do PhD in data science? And I am in MSc graduate in mathematics. Yeah, all the mathematicians having the programming background, or they can learn this programming using R or Python. MATLAB is there, Mathematica is there, but Python and R programming is highly used for the data science. So you can become, or you can do PhD in data science because you have mathematical background. So I think all the things which are presented in the session are very quite interesting and informative. And still, if there is any question you want to know more about, please feel free. Either it is time to also close the session.
Hello, sir. Hello, sir.
Yeah.
Hello, sir. I hope you are going to have the recordings of this sent to us.
Yes. Also, the feedback form has been shared in the chat box. You can submit your feedback, and we will provide you the cloud recording of this webinar. You can attend in the future. You can use it. Okay. So I will send you. Yes, I will send you the email to all the participants. There are many participants who are not able to attend this webinar due to the time difference, because most of the participants are from Ethiopia and Nigeria, because the time difference is about 6 hours, about 5 hours with Nigeria. Okay.
Yes.
So don't worry about that. We will send the recording link to all the participants through their email address. Okay. And as along with the feedback form.
Okay. Then the last question is, the last Saturday's training, on Excel. There was a training on Excel last Saturday.
And last Saturday on Excel?
Excel, yes.
Okay.
I made a request.
You got your certificate or not?
I have it. I have got it. I got the certificate.
Okay. So
now, I made a request. I made a request that can we have a PowerPoint training too?
Yeah, we will provide you in the very coming days. In the month of August, there will be several programs will be organized on Excel, on R programming, and Python also.
Okay. They're on PowerPoint. On PowerPoint.
Yes, PowerPoint also. PowerPoint is very quite easy, and still, if there is required, we will organize one session for the PowerPoint, because this is also very highly used to present, because there are sometimes we need animated. So we will organize one session for the same.
All right. Thank you very much, sir. We are very grateful. We appreciate this. And God bless you.
Thank you.
You're welcome, sir.
Just 1 minute, sir. Are you there?
Yes, yes, yeah.
Just 1 minute, sir. Please wait. Okay. So in the present session, all the areas which we discuss, proposed for the webinar has been discussed, and all type of questions from the audience is also has been discussed. And we all very thankful to share your valuable and informative thought in the present session. We present a certificate of appreciation to Dr. Parveen K. Gotura for imparting his valuable and informative thoughts on one-day international webinar on basic bioinformatic operations using Python. And I hope all the participants enjoy this session and got a lot of information for their respective area. And some are there participants who are working in mathematics or background is mathematical, but still they can move for bioinformatic or biomathematical modeling, and they can start their career in also bioinformatics, but need some basic background related to the biology or biotechnology, and still then they can work, because there are so many colleagues from different countries who are working on bioinformatic or biomathematical modeling or biostate, but the background is mathematics, and this is a very emerging area and a lot of scope also. Thank you, dear professor, for your valuable time for this session. This session was very informative, valuable, and knowledgeable to all the participants. I also enjoyed this session, and also thanks to all the participants who joined this meeting from different countries to spare your valuable time. And at the last, I would like to ask from all of you, please open your webcam so that we can have screenshot for this webinar. Please open your webcam. I request to all the participants to open the webcam.
How do I open the webcam? There?
Yes, please.
How do I open the webcam?
At the bottom, you can see your video, mute, unmute, and there is a video stop and open.
Just click on your video.
Supposed to be a singer. I'm nervous is smiling.
I've seen you.
Good morning, man.
Patience, Ivan, and Favor, you both are the same place. Yeah.
Yeah, from Nigeria.
Yes, I know you are regularly attending our webinar, seminar, and workshop. Many events you have attended.
Mommy smile, come now.
I request to all the participants.
Can you see
there
now?
are many participants who did not open their webcam.
Can you see?
Yeah, I can see.
No, what should I listen to?
Okay. So,
Thanks to all and have a nice day. Now it is time to close this meeting.
I can even see a colleague of.
Yeah. Feedback
Thank you
form,
too.
feedback form has been shared on your chat box. You can submit. Because after submitting the feedback, we will send your certificate.
Thank you very much. we appreciate you.
Hello.
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 Hello, please. Hello, Odesina.
Yes, I just want to say thank you for this lecture. It's really an eye-opener. Really.
Can you please repeat your question? Your voice is breaking. Can you put your question in the chat box?
Put your question on the chat box. Chat box. You can put your question on the chat box. No. Jeff. Must I call you? Your thing even ring, your phone ring, Randy.
Okay, Odesina, thank you for your nice word. Please don't forget to submit your feedback form. Okay. And also I request to all the participants who are attending this webinar on YouTube live streaming, the link for the feedback is also shared in the chat box on the live streaming on YouTube.
Good. Hello, Prof. Hello, Prof.
Hello.
Please, I don't know. We will have to have a collaboration with you with time. I don't know how you're going to assist us.
Yeah, you can collaborate with us.
Yes, we need. I'm from University of Ibadan, Nigeria. And I belong to a professional body that you can help us. Especially in the area of training. Are you with me?
Yes, please let me know. Yeah.
So I said, I'm with the University of Ibadan in Nigeria.
Okay.
And that I belong to a group of professionals. I'm a technologist by profession. And I would like you to, collaborate with you in the area of training.
Yeah, you are most welcome. You can collaborate with us. And also I would like to invite further collaborations of various universities from Nigeria, if they are willing, because we are trying to collaborate with the universities, institutes. Okay, we have several MOU has been signed recently. There are 2 universities in Ethiopia and 2 universities in India and 1 institute, and several are in the process. They wanted to collaborate with us, and we are also willing to invite several resource persons and who are expert in their respective area. If they can contribute for the society, you are always most welcome. You can send your brief introduction or CV at my email address. I am sharing in the chat box my email address. You can see.
Thank you.
Yeah, I am sharing. Right. You can.
All right. So what I will do is I will speak with my boss, and then we will have a way of getting in touch with you.
Yes. You can ask for to sign the MOU, Memorandum of Understanding, and after signing this MOU, there is a proposal. First, you need to invitation to us for the signing the MOU, and then after we will provide some template. There is a very small formality of documentation, and once we have signed the MOU, then there are a lot of scope to organize several type of programs in local institute as well, virtually also. And this will be very beneficial for the institute as well as for the faculty members and the students, because we have a very large group of resource persons, experts, and to provide different type of trainings in the different areas of program, like R programming, Python, Mathematica, Maple. Okay, and all these are very, there are several applications in different areas of science, engineering, for different to sort out different type of problems. Okay, so there is no problem, and once after collaborating, we can go ahead and we can help each other in various ways are there.
Okay.
Yeah.
Thank you very much.
Thank you, Professor. Thank you. And please,
I'm not a professor. I'm not a professor.
Pardon?
I said I am not a professor.
Okay.
Yes.
Sorry, in outside the country, they call every lecturer a professor. They call us professor. That is the title they give lecturers, not necessarily that you are a professor.
Okay.
Good patience, Evan. You handle my question.
Thank you.
Thank you, all of you. Have a nice day. I have shared my email address on the chat box.
Yes.
Thank you.
Yes. Thank you too. Have a nice day.
Have a nice day. Thank you all of you. Now we are ending.